name is Siti Hanisa binti Baharudin. My supervisor is Dr. Tun Nurul Aimi binti Mak Jaafar. My final year project title is Levels of CO1 Divergence in genus Scomberomorus, Perciformes Scombridae. Scomberomorus is a genus of ray fin bony fish that contain 80 species range worldwide in tropical to temperate latitudes. Most species are coastal epipelagic, and species has morphology confused and difficult to recognize morphology differences. So with DNA barcoding, species ability to identify or to analyze from the tissue level. DNA barcoding is a taxonomic method to identify species and best solutions when morphology is of limited use. Lead to misidentification when two species are morphologically very similar and the data uploaded in GenBank by non-taxonomists. Aim of my researcher are to determine the levels of CO1 divergence in genus Scomberomorus and to test the accuracy of species identification in GenBank. There are 80 species genus Scomberomorus was collected from fish base, and 280 sequence of 12 species has been retrieved from GenBank. 280 sequences were aligned and neighborgeometry were constructed. The sequences were aligned using muscle algorithm, which was implemented in Megaseq, and the cumulative parameter distance measure was employed. 280 CO1 barcode sequences representing 12 different species of genus Scomberomorus were obtained from GenBank. Scomberomorus syrah has the lowest number of sequences, while Scomberomorus commerson has the most number of sequences. The mean intraspecific of 280 CO1 sequence K2P distance was 1.37%, and the lowest distance was 0% observed in Scomberomorus plurinatus with 4 individuals, while the highest was 0.8%, both Scomberomorus regalis with 60 individuals and Scomberomorus commerson with 60 individuals. Scomberomorus regalis show deep divergence with maximum nucleotide divergence of 3.2%. A potential 47 specimens have been detected as potential misidentification. Phylogenetic analysis show that Scomberomorus guttatus form 3 distinct clades and mix with Scomberomorus plurinatus. Clade 1, both of them showing similar geographic range, which from Bangladesh. Clade 2 consists of Scomberomorus guttatus, and Clade 3 consists of Scomberomorus plurinatus and Scomberomorus guttatus. These clades are defined by some modes of geography. Individual of Scomberomorus regalis with genetic divergence 3.2% were grouped with Scomberomorus brasiliensis. Others 50 specimens of Scomberomorus regalis were grouped together, contained with the cluster of Scomberomorus maculatus. They have combined characteristics. Deep divergence detected among individuals of Scomberomorus commerson. The cluster were separated by 3.2% nucleotide with high bootstrap value 98% to 100%. This study confirmed that taxonomic uncertainties do occur among species within genus Scomberomorus. Accuracy sequence deposited in GenBank are important to avoid ambiguous results.